Hello students. While doing the topic potentiometer, we had talked about primary cells. I told you that primary cells are cells which cannot be recharged. We have another type of cells called the secondary cells which can be recharged, such as the cells we use in our car batteries, scooter batteries, in the inverter in our homes. These can be recharged. But the primary cells, they cannot be recharged. An example of the primary cell is a simple dry cell, the small cell you use in your clocks or torch, etc. They cannot be recharged. Such cells are called as primary cells. The history of primary cells is very old, and the first primary cell probably was the voltaic cell. While doing the experiment for potentiometer, we will be using two freshly prepared cells in the lab, the Daniell cell and the Leclanché cell. I will explain their construction to you one by one. First, let us see the Leclanché cell. The Leclanché cell is made of a glass container. There is a glass container. You can have a plastic container as well. This is a glass container, glass jar. The electrolyte that is used is a NH4Cl, ammonium chloride. So, freshly prepared solution of ammonium chloride is prepared and put in the cell. Besides this, a porous pot, a cylindrical white element we have, which is the porous pot, and it is closed from its top. This porous pot, it has a mixture of MnO2 and carbon inside it. And besides this mixture, it has a carbon rod fixed inside it. This is a closed porous pot. What is meant by a porous pot? A porous pot is just like your matka. The walls of the pot, they have pores. These pores enable the movement of the ions through the porous pot. The ions can move in and out of the porous pot. Besides this, there is one more electrode that is used. A zinc rod is used. The zinc rod is the negative terminal and the carbon rod is the positive terminal. In this way, in the lab we prepare a fresh Leclanché cell. The another cell that is used to compare EMFs of two primary cells is the Daniell cell. The Daniell cell uses a copper container. The copper container, it itself acts as one of the electrodes. So we have a terminal here, and this acts as the electrode, and this acts as the positive electrode. Inside this, a freshly prepared solution of copper sulfate is taken. A porous pot is placed here. This porous pot is open. In the Leclanché cell, it was sealed. It was already packed. But here, in this Daniell cell, this porous pot is open, and inside the porous pot, we prepare a dilute solution of H2SO4 and put in the porous pot. Inside the porous pot, we insert a zinc rod. This zinc rod acts as the negative electrode. These are the different components of the cell, and we can prepare them in the cell while we are performing the experiment for potentiometer to compare the EMFs of two primary cells, or if we want to find the internal resistance of a cell, we can prepare the Leclanche cell or the Daniell cell and find their internal resistance. Thanks.